Good evening, one and all. Myself, Dr. Shrey Bodhankar, working as Assistant Professor at School of Agriculture Sciences, Department of Microbiology, Anurag University, Hyderabad. And my topic of presentation is on role of endophytes in management of abiotic and biotic stress. So let me begin my topic with the climate change. Climate change has become real and tangible, affecting people's life worldwide. Over the past decade, it has become warmer, polar ice caps are melting, sea level is rising, and these extreme events are becoming more frequent and more intense. According to the United Nations frameworks, there will be a surface rise in temperature of 4 degrees Celsius by 2100, and there will be a 30% decline in the yield of various crops. Among different abiotic stresses, drought is one of the major stress which is impacting the agricultural productivity worldwide. If you see in India, 46% of the agricultural land is irrigated and rest 54% of the agricultural land rely on the rainwater for the crop growth. The available record suggests that the rain-fed tract experience 3 to 4 droughts for every 10 years, of which 2 to 3 drought are of moderate intensity and one will be severe. So what are abiotic stresses? Abiotic stresses includes high temperature, cold, drought condition, salinity, and metal toxicity, overflooding to the different crops. Whereas biotic effect of different pathogens on the crop. Also, you have and the insect pest. So these are categorized into biotic stresses. So when you see the biotic and abiotic stresses, which affects the plant of all growth of the plant and the production, particularly the reproductive stage of the crop. Of course, we have the important strategies such as breeding and using of molecular tools to combat the abiotic and biotic stress. But these are cost intensive and time consuming. We also have other problem apart from abiotic and biotic stress that is which is a global problem that is increasing in the population. If you see here, the current world population is around 7.9 billion and it is expected to raise 10 billion in next 50 years. So we need a huge food for growing population. How will we provide the food for the growing population? Of course, we have different strategies such as increasing the agricultural land, but we cannot do that because we have limited resources. We can also use farm mechanization, but we cannot do intensively because again it's an expensive process. Of course, we can also use chemical fertilizers and pesticides. That's what we are doing today or these days. But again, it is against to the environment and human health. We can also use the transgenic crops or genetically modified crops, but it is already restricted in various countries. The possible solution will be use of plant-associated microorganisms. As you see here, microorganisms are present everywhere in the marine environment, in the desert soil, or in the fish, in eggs, honeybees, in different trees, flowers. So the diversity of the microorganism is enormous. So far, only 1% of the microorganisms have been known in the world today. Most of the researchers or the scientists are focused on rhizosphere microorganisms in the past decade. Rhizosphere microorganisms are the bacteria or the fungi which are living in the vicinity of the root zone, which play a major role in protecting the plant against abiotic and biotic stresses. We call these bacteria as plant growth promoting rhizobacteria. The example of these bacteria are Bacillus, Pseudomonas, Azotobacter, Azospirillum, Rhizobium. Recently, researchers and the scientists are working on a bacteria called or microorganisms called endophytes. So what are these endophytes? According to the definition given by Bacon and White, these endophytes are the microbes that colonize living internal tissues of the plant without causing any immediate overt negative effects. A true endophyte should able to infect the plant and establish in the internal tissues of the disinfected plant from which is previously isolated from it. You can find the endophytes in the plant in various parts such as seeds, roots, stem, leaves, and pollen grains. These microbes exhibit an array of beneficial properties that are similar to plant growth promoting rhizobacteria, what we call it as PGPR. Their localization inside the plant give them advantage over the rhizosphere microbes in terms of their availability of nutrients and the shield against external unfavorable conditions like high temperature, salinity, metal toxicity, and overflooding. Endophytic bacteria might stimulate the plant growth by producing certain phytohormones, bioactive compounds, growth regulators, enhancing the host anabolism, or by regulating the phytohormone levels, similar to plant growth promoting rhizobacteria. These endophytic macroorganisms having more intimate relationship with the host plant may prove to be more promising than the rhizosphere microbes. Among the endophytes, seed endophytes are of great interest because their capability to colonize the developing seeds, stay latent in the dormant seed, and transmit to the next generation after generation in the plant development. These endophytic bacteria, isolated from the seeds, have been found to produce certain antimicrobial compounds, phytohormones, enzymes, secondary metabolites that make them industrially as well as agriculturally very important. The application of these endophytes have been reported to improve the plant growth and physiological process under abiotic and biotic stress condition. As you can see in this slide, which depicts the passing of the endophyte from the seed to the stem, then to the root, then followed by the fourth figures again to the seed. These are the different endophytes which have been reported in different crops. Burkholderia phytomerans, which has been isolated from grapevine. Orthobacter bacillus has been isolated from sweet pepper. Perimorphospora indica, which has been isolated from barley. Trichoderma, which has been isolated from cocoa, and Bacillus lyconiformis, Bacillus subtilis, and Bacillus subtilis 78 and 72 have been isolated from maize. This is the work which I have carried out at Indian Council of Agriculture Research at institute called CREDA, Central Research Institute for Dryland Agriculture, from the seeds. Now, when I say endophytes, we need to look how the endophytes get an entry into the particular plant system. So endophytes may get entry into the host plant via rhizosphere, that is through branching sites, cracks, or the wounds, and through the philosphere, that is including the stem. It can be through the cracks or the wounds, into the leaves via stomato, and flowers through the pollen grains. Root exudates play a key role in attracting or recruiting these soil microbial communities in the root zone, facilitating host microbial crosstalk and endophytic colonization in the plant system. And if you can see their importance, it is very much similar to the rhizosphere plant growth promoting rhizobacteria, which helps in root branching, number and length of the lateral roots. It will increase the lateral root hairs, which forms the biofilm on the surface of the root, which helps in protecting under the high temperature and drought stress condition. It also helps in anchorage or support in the soil. It forms the soil aggregates, which increases the physical structure of the soil. It helps in biological nitrogen fixation, solubilization of nutrients such as phosphorus, zinc. It also helps in biocontrol of different soil-borne pathogens. It can be a bacterial pathogens or fungal pathogens. It is also resistant against various abiotic stresses. This is the practical evidence of isolation of endophytic bacteria, which was carried out at ICR CREDA, how the endophytes are isolated. So this is, for example, to understand the audience. First, we have taken the seeds. These are the maize seed, which has been treated with mercury chloride to eliminate the surface flora which is associated with the seed. Then the seeds were imbibed in a sterile distilled water to just to soften the seeds. Later, the seeds were crushed in the mortar and pestle, which is a sterile mortar and pestle, and the aliquot of the seed is added to the Petri plate. On top of the Petri plate, different media based on the research interest or the scientific interest, one can choose a different media for the isolation. So here we have used TSA media for the fast growers and R2A media, reasoners to A agar media for the slow growers. And two of the seeds we just placed to see whether there is a real any contamination or not. And two of the plates we kept as a control. When I say the endophyte, it means it need to be re-isolated from the plant system once we seed treat it. So this is another experimental evidence where the seeds were treated with rifampicin marker, resistant marker. The endophytic bacteria was mutated and seed treated. Then again, it has been isolated from different parts of the plant, such as root, stem, and the leaf, just to track the endophytes from the plant system. After isolating the endophytic bacteria, we have seen the morphological characterization or morphology of the bacteria, that is based on the form, whether it is circular, irregular, filamentous, or rhizoid, or whether it based on the elevation, whether it is raised, convex, flat, uh, or any other shape. Based on the margin, we also characterized them into entire or lobate based on the margin of the different bacteria. So after the characterization, morphological characterization, we have done certain biochemical tests using a kit which has different biochemical substrates ranging from 1 to 35. So initially the endophytic bacterial culture is raised overnight, that is for 12 to 24 hours. After the inoculation, the results have been recorded based on the color change of the substrate. To characterize the bacteria molecularly, we have isolated the genomic DNA of endophytic bacteria followed by polymerase chain reaction. And here, if the cultures are more than 100, we have done ADRA procedure, amplified DNA restriction enzyme analysis. Based on the pattern of restriction enzyme, we have grouped the different endophytic bacteria and selected few of them and given for the sequencing. After the sequencing, this is the growth, this is the phylogenetic tree which has been generated, which shows different bacteria. Majority of them are Bacillus subtilis, which has been isolated from maize seeds. And to check whether the endophyte is really potent against different abiotic stresses like drought, temperature, and salinity, these endophytic bacteria are exposed to polyethylene glycol 6000. PEG 6000 is a solution which mimics as a drought situation in the laboratory. So we have exposed the endophytic bacteria at different concentration ranging from 10% to 40% to see the growth of the bacteria under the water deficit condition. Similarly, to know whether the bacteria is tolerant to high temperature, the bacteria is exposed to different temperature range ranging from 30 to 60 degrees in incubator for about 2 to 4 days. Later, to know whether the bacteria is really tolerant to high saline conditions, these bacteria were exposed to salinity, that is sodium chloride, ranging from 2 to 4%. And you can see here on the Petri plates, these bacteria have been spot inoculated on respective media and incubated at different conditions. The in vitro screening of the bacteria was carried out to know their plant growth promoting traits, that is, production of ammonia, production of siderophores, production of phytohormones, and their nutrient solubilization, which includes phosphorus solubilization, zinc solubilization, and potash solubilization. I've also tested the endophytic bacteria against soil borne pathogens, particularly in maize. They are Rhizoctonia solani, Sclerotium ralsii, and Macrophomina phaseolini. As you can see in this picture, this is the siderophore production, extreme left side picture. These bacteria are producing siderophores, and you can see these are antibiotic resistant bacteria. These are the lysozyme enzyme producing bacteria. Then extreme right picture, which depicts the EPS producing bacteria, that is exopolysaccharide production. And these bacteria also tested against different enzymatic production, which includes lipase, esterase, amylase, pectinase, cellulase, and protease. Why using? Why we need to select the lytic enzyme? Because most of the lytic enzymes helps in combating or fighting against the fungal pathogens, or it also helps in degrading the cell wall of the plant cell, which helps the endophytic bacteria gain an entry into the plant system. So this is the published research article, that is maize seed endophytic bacteria, their antagonistic lytic enzyme production, in 3 Biotic journal. I've also tested the endophytic bacteria in its different solubilization, that is phosphorus solubilization, zinc solubilization, and potassium solubilization, by spot inoculating the endophytic bacteria on respective media containing phosphorus, zinc, and potassium. And you can see the zone of inhibition, the clear zone around the colony. After in vitro studies, the farmers required endophytic bacteria which need to be performed well under the field conditions. So we have tested the endophytes by doing the screening in the pots. The pot screening is categorized into 3 types. Initially, we have screened in a primary screening was done in a small size pots. Secondary screening was done in a medium size pots. And lastly, the last screening was carried out in a large size pots. So here you can see the small size pots contains approximately 800 grams of soil, where we have seed treated the maize seed with the different endophytes. Replicates were maintained. In this primary screening, we have screened around 110 different bacteria, and the observations were made on their chlorophyll content, shoot length, shoot fresh weight, shoot dry weight, root fresh weight, root dry weight, in comparison with the control. Here, the secondary screening was carried out in a medium sized pots, which includes nearly 4 kgs of soil. And the observations were again made on chlorophyll, shoot length, shoot fresh weight, shoot dry weight, root dry weight, relative water content, proline, sugar, soil moisture content. And the last screening was carried out in a large size pots, which includes nearly 10 kgs of soil. Here we have chosen limited bacteria or the potent or well performing bacteria, and the observations were made on chlorophyll, shoot length, shoot fresh weight, shoot dry weight, root fresh weight, root dry weight, relative water content, proline, sugar, soil moisture content. Finally, among 110 bacteria or endophytic bacteria, I have chosen 11 different isolates and did an experiment under the field condition at ICAR TRIDA, where we have a gunical research farm. Here, after sowing the maize crop, the observations were recorded, similar observations, that is chlorophyll, shoot length, shoot fresh weight, dry weight, root fresh weight, dry weight, relative water content, soil moisture content at different intervals. That is at 21 days after sowing, 48 days after sowing, and 74 days after sowing during the dry spell, or I can say during the drought condition, natural drought condition. And we have also recorded the yield parameters. Apart from field studies, I have also focused on the possible mechanisms behind the microbial induced stress tolerance. So, for that, I have conducted a growth chamber experiment to study their mechanisms behind the drought tolerance. And here, the observations were made on plant physiological properties as well as plant growth properties like shoot length, shoot fresh weight, dry weight, chlorophyll, and antioxidant status. Endophytes regulate drought responsive genes. So we have also isolated certain, we have also isolated the RNA and studied their expression levels. As you can see here, this is the 2D gel electrophoresis pictures, which depicts the protein profile of the endophytes in the maize leaves. So these are the different drought responsive genes: dehydrin, ZM, GRM, ZM2G, 5584, which has been decreased under the drought stress, and dehydrin has been increased under the drought situation. So we also have other endophytic fungus called trichoderma. As you can see in this picture, Trichoderma harzianum. This is from ICAR, IAOR, Indian Institute of Oilseed Research, where they are using this as a formulation to treat soil-borne pathogens. So the recommended dosage is 5 to 10 grams per kg of seed to eliminate the root rot, stem rot, and leaf spot. It is a biocontrol fungus. Endophytes can act as a biofertilizers, which helps in nitrogen fixation, phosphorus solubilization, phosphorus mobilization, potassium solubilization, zinc solubilization, as it has similar properties like plant growth promoting rhizobacteria. It can also be used in decomposing the waste material generated from the agriculture sector or the agriculture field. These endophytes, you can see here azotobacter or azospirillum, which are free living nitrogen fixing bacteria, can also be an endophyte. It can be used in all crops. Azotobacter can fix around 15 to 20 kgs of nitrogen per hectare. Azospirillum can fix around 20 to 25 kgs of nitrogen per hectare. We also have uh, other endophytic bacteria, but it is very much restricted to the root zone, which is known as VAM, vesicular arbuscular mycorrhizae, which is helping in phosphorus mobilizing uh, bacteria. It's a fungus which stays in association with the plant roots and spreads and penetrates into the root system. It also mobilizes the potassium, zinc in a required amounts to the plant. It also protects the plant, increases the root architecture, as you can see in the picture, the left one. With, it also holds the water under the drought situation, controls the nematodes, and has the biocontrol activity. Of course, as you can see here, decomposing biofertilizers, which are used nowadays instead of stubble burning. So after the harvest, stubble burning, it kills the microorganisms and friendly insects. To avoid that, one can use the biofertilizer, uh, which can decompose the agriculture materials. But so far, there are very less research has been carried out on endophytic bacteria, which can decompose the plant material. So there are certain cellulomonas bacteria, trichoderma, pseudomonas, bacillus, which can be an endophyte. Agriculture waste can be turned into a compost and can used as a biofertilizer. It, we can avoid burning of stubble better to make a compost and use as a eco-friendly biofertilizer. So this is already available in the market, but this is not an endophytic one. Most of the researchers are targeting on the endophytes, which can further used as a biofertilizer after decomposition of the plant material. How to use the biofertilizer or endophytes or the bioagents? It can be used in many ways for the seed treatment, root dipping before transplantation, direct application in the soil. It can be applied in the drip. So biofertilizers are available in various forms, mostly in the powder as well as in liquid or in granular form. So the powder form of the biofertilizers, it can be a talc or lignite based formulation. When you see their viability or shelf life, the viability of the powdered based formulation will be for 6 months if you are storing at 25 to 30 degrees, and it can cost you around 40 or 50 rupees. If it is a liquid formulation, the shelf life will be around 1 to 2 years if one is storing around 25 to 40 degrees. The price ranges from 300 to 400 per liter. One should not treat the chemical fertilizers. If you are using the bio-based inoculants, it can be endophytes or rhizosphere microbes. It is better to avoid. Note, if you are using any chemicals, first treat with fungicides, then with chemical fertilizers, then treat with a bio-based formulation. If you see the dosage of liquid biofertilizers, 200 ml per acre. For seed treatment, use 200 ml of bioinoculant or biofertilizer to treat 1 acre of seeds. If you are using talc or lignite based formulation, which is a powder based formulation, you can use 350 grams per acre, that is 5 to 10 grams per kg of a seed. For seed treatment, you can use powdered biofertilizer to treat 1 acre of seeds. If you are going for a root dipping, you can add 20 ml of bioinoculant into a 1 liter of water and use it for root dipping. Soak it for 20 minutes to half an hour and go for transplanting immediately. If you are using as a soil application, take 200 ml of bioinoculant, add to 100 kg of vermicompost or farmyard manure, keep it for 10 to 15 days, that is to enrich with the microbe. Apply before plowing and see that it should contain a moisture. It should be plowed before sowing. You can also use through the drip irrigation by adding 20 ml of biofertilizers to 100 liter of water. This is the schematic representation or the roadmap of isolation of biofertilizer or rhizosphere microorganisms, where one can select the first picture says the rhizosphere. One can choose a plant material, either it can be a seed or root, stem or a leaf. One can isolate the bacteria or the fungi, then do the in vitro studies. Later, one can go for the greenhouse screening, followed by field evaluation. Then there are certain then product development, followed by you have regulatory approval and certification. Then you can release as a product into the market. So if you can see the biostimulant market potential, it is expanding at a robust rate during 2023 to 2030. If you see, the market size is projected to increase 307 million to in 2023 to 849 million by 2030. Compound annual growth interest CAGR of 15.61%. In India, the Cabinet Committee on Economic Affairs has released a budget in the name of PM Pranam, that is PM Program for Restoration, Awareness, Generation, Nourishment, and Amelioration of Mother Earth scheme for about 3 lakh 70 crores, 128 crores for biostimulant market. Thank you. I would like to thank agricultureinformation.com for giving me this opportunity. Thank you, one and all. Yeah.
I'll now take up some questions.
Oh, sure.
Okay. Yeah. So first question is, how
Yes.
do endophytes help plants withstand environmental challenges like drought and extreme temperatures?
Okay. So endophytes has the capacity to produce certain metabolites uh, under the drought situation. By these metabolites, like secondary metabolites, what we call. So these metabolites helps the plant to sustain under the stress condition, as you say, the drought, salinity, or high temperature.
Okay. Yeah. So, and can you provide examples of specific plants or crops where endophytes have shown remarkable effectiveness in improving stress tolerance?
Oh, you mean the example of the endophytic bacteria, which has really helped under the drought stress?
Yes.
Yes, you can see an example here, which is in slide number 13. This is
Your
from my.
screen, your sorry,
Oh, okay.
your PPT is, yeah.
Let me share, ma'am. Just a second. So if you can see slide number 13, the last one, Bacillus licheniformis, MSEB 17, and Bacillus subtilis, MSEB 78 and 72. So these are the endophytic bacteria which I have isolated from the maize seed, and these are performing better under the field conditions. So these are the field maize seed endophytes. The name of the bacteria is Bacillus licheniformis, or the example of the bacteria is Bacillus licheniformis, Bacillus subtilis. MSCB is the code name, that is maize seed endophytic bacteria. 17 and 78 or 72 are the number of the isolate. And this we have published in 2017 and 19 in international journals. It has improved the chlorophyll content, plant biomass, proline content of the leaf, relative water content, and also finally yield of the maize crop. Okay, ma'am. Okay, fine. Yeah, sure.
Yeah, Mr. Shrey,
Is yes, ma'am. Is it
technical
audible
problem.
now?
I think. Yeah, you're audible now.
Okay. So as you asked me that, are there any real endophytes which have been helping the plant under the drought stress? Right. This is the question. Hello. Hello. Hello.
That's
Is
the
it
question.
audible? Okay. Yeah. So as you can see here in slide number 13, Bacillus licheniformis, the last one, MSEB 17, Bacillus subtilis, MSEB 78, and 72. These are the three different bacteria which I have isolated during my PhD study. That is from the maize seed, and it helped under the drought condition. It also increased the chlorophyll content of the leaf, biomass of the plant, leaf proline content, total sugar content, and the soil moisture content. Apart from that, it also increased the yield of the maize, and we have published this in two international journals in 2017 and 2019.
Okay.
Yeah.
Yeah. So next question is what?
Yes.
What are some of the common biotic stressors that endophytes can effectively combat, and how do they contribute to the plant's natural defense mechanisms?
Okay. So you can see here. Just a minute. So you can see here, endophytes can combat the most common stresses like drought, high temperature, low temperature in general. Uh, it also helps cope up with the plant system, because plant also has the genome. So it will better communicate with the genetic material which is present in the plant system, as well as the microbe genetic material. Based on this, it will release certain proteins which helps in combating different stresses. You can name it as abiotic stresses like drought, temperature, salinity, or abiotic stresses, or biotic stresses, that is fighting against different fungal pathogens.
Okay. And how do farmers and agriculture practitioners
Yes, ma'am.
benefit from incorporating endophytes in their crop management practices, particularly in terms of reduced reliance on chemical interventions?
Okay. So as I told you, most of the researchers are focusing on endophytes. Earlier, most of the scientific researchers have been isolated the rhizosphere microorganism. The practice will be similar as of rhizosphere microbe. Only thing, here you can differentiate between the rhizosphere microorganism as well as the endophyte. The endophyte has an upper hand in protecting against various stresses than the rhizosphere. Why? Because it is closely associated with the plant system, and farmers can use the same technique what they are using for the rhizosphere. That is by they can use for the seed treatment, root dipping, direct application into the soil, or the drip irrigation, because these formulation will be available. You can see as a powdered formulation or a liquid formulation.
Okay. Next question. What are some practical methods for farmers to encourage the proliferation, sorry,
Yeah.
proliferation of beneficial endophytes within their crops without disrupting the existing ecosystem?
Okay. See, it will not harm. It will not see. First of all, endophytes are natural biological molecules. It will never ever harm to the ecosystem. So farmer can use in his crops, provided one should maintain. Like if they are using a chemical fertilizer, one should take up a small plot which is far away from the chemical fertilizer or chemical farming, then he can create a small plot and sow the crop over there and maintain till they harvest.
Okay. And are there any notable differences in the efficacy of the endophytes in managing stress between different plant species? And if so, how can
Yes.
farmers identify the most suitable endophytes for their crops?
Of course. So in the market, as you can see, there are so many formulations. Of course, there are so many fake formulations also. So a farmer can take up the few of the formulations from different companies. There are notable formulation, as I told you, Perimorphospora indica, which is a suitable endophyte, which is performing better than the rhizosphere organisms. So farmer can take up the trials. I mean to say the experimental trial in their own farms in a small plots and see the difference, how well it is performing in terms of the yield or in terms of the fodder.
Okay. And how can the use of endophytes contribute
Yes,
to the overall sustainability and long-term health of agricultural systems? And
yes.
what are the environmental benefits associated with their application?
The environmental benefit will be like it will increases the biodiversity which is present in the soil. These endophytes has the capability to pass the endophytes from one generation to the next generation crop. As you see, if you are sowing a seed in this year or the first in Rabi or Kharif season, once you harvest the crop, you may have the endophyte in that particular seed again, the harvested seed, which you can again resow it and use it or pass it to the next generation. That is the power of an endophyte when you compare with the rhizosphere.
Okay.
Yes, ma'am.
Hmm. Okay. And are there any specific, yeah, are there any
Uh,
specific challenges or limitations that farmers should be aware when
yes.
utilizing endophytes for stress
Okay.
management? And
Okay.
how can these challenges be addressed effectively?
Challenges are there. The main challenge is the shelf life. Most of the researchers are targeting to improve the shelf life. So, a farmer who is purchasing a biofertilizer, he has to, in a powdered formulation, they have to finish it within 6 months. So, that is one kind of limitation. Other limitation will be like they need to use the gloves, which can again cross contaminate with other sources, or they might contaminate the microbial population which is already provided with the powder. And the other challenge is in maintaining at the right temperature for storing these bio-based formulations.
Okay. Hmm.
And the another challenge is that a farmer should not expect that if you are treating with the endophyte or any bio-based formulation immediate effects, because any organic thing will take time. So the farmer should have the patience to wait at least for 2 years to get the returns.
Hmm. Hmm. Okay.
Yes.
And can you share any success stories or case studies where the integration of endophytes has led
Yes.
to significant improvements in crop
Yes.
resilience and overall farm productivity?
Okay, so one example I can quote you, I was associated with Indian Institute of Oil Research for implementing or promoting so-called endophytes in a farmer's field. So there we have taken different experimental plots, that is from the farmer itself, across ranging from 5 acres to 10 acres. In the crop is a groundnut crop. They have successfully got a good results in their field. It helps in combating the fungal pathogens as well as the drought stress. This study was conducted at Mahbub Nagar district of Telangana.
Okay. Yeah. Um, Yeah, can you share any success stories? I think, okay, sorry, you have already answered. What
Yes.
recommendations would you offer to farmers interested in incorporating endophytes based strategies in their agriculture practices, and how can they ensure a smooth and successful transition to this approach?
Okay. Initially, farmers should start with a small area of land instead of going directly in acres together. So they can have an experimental plot for about 1 or 2 acres if they want to see the real impact comparatively with the chemical farming. The first thing I can recommend is seed treatment. This is the easiest and the best method a farmer can practice. Apart from that, if they are going for a paddy, the next method of treatment will be the root dipping, and if for the horticultural crops, I can say the direct application to the soil or a drip irrigation.
Okay.
Yes, ma'am.
Yeah. Okay, Mr. Shrey, I don't see any more questions from our participants. So
All
now
right,
we
ma'am.
have come to end of question round.
Okay,
On
ma'am.
behalf of agricultureinformation.com, we'd like to thank you
Thank
for
you,
the
ma'am. I
detailed
thank you.
presentation and answering all the questions in depth. And we
Fine,
also
ma'am.
like to thank all the participants for joining this meeting. The meeting
Thank
will
you.
now be closed. Have a
Okay.
good and safe weekend ahead.
Thank you. Thank you, ma'am. Thank you
You're
for
most
giving me
welcome.
the. Thank you.
You're most welcome. Thank you.